we can start the class now.
वही व्हाट्सएप ग्रुप नहीं
We
है।
are going to study. We are going to study beta HCG. This is the new experiment on you in your practical. Beta HCG क्या होता है? हम इसके बारे में जानेंगे. We study the procedure, the principle, the significance. Here HCG, the full form is S for human, C for chorionic, and G for gonadotropin. So HCG is a human chorionic gonadotropin. This is a hormone. One of the example of the hormone. This test is also known as pregnancy test. And the aim of the experiments is to detect beta HCG in urine sample. Basically, I have taken a procedure to detect beta HCG in urine sample. So the procedure is based on detecting beta HCG in the urine sample only. We can do serum sample also. We can use serum sample, but most preferably we can try with the urine sample because it is very convenient to collect urine sample. Though the blood is somehow invasive method, so urine method can be preferred. Okay, urine collection can be done. And the method is based on rapid chromatographic immunoassay. Earlier, so many tests we have discussed, so many tests we have learned. So those tests were based on the ELISA. This is the only test we are going to study, which is based on the rapid chromatographic immunoassay. We have used the term rapid because it can give the result very rapidly. Means very soon you will get the result. And the chromatographic means it's a based on the chromatographic procedure, where there is a movement of the antigen or what we call it a hormone here. So the hormones move through the capillary motion, and it binds with a specific antibody against the these hormones. And here we have used immunoassay because the reaction is like antigen antibody reactions. Okay, so the method is called as a rapid chromatographic immunoassay. Okay, so we have used two method. We have compiled two method. One is a chromatography technique and immunoassay technique. So both of the technique has been used to perform the HCG test. What is the purpose of beta HCG test? So this test is done for the qualitative detection of human chorionic gonadotropin in urine to aid in the early detection of pregnancy. So if anyone, the female wants to know that either she is pregnant or not, and then she can go for this test. And this test gives us the early detection of the pregnancy. Let's know the introduction of this test. HCG is a glycoprotein hormone. Chemically, the HCG, the human chorionic gonadotropin, it is made up of glucose moieties and protein moieties. So it's a glycoprotein hormone, and generally this hormone is produced during placenta, produced by the placenta during pregnancy. Normally, HCG can be detected in both urine and serum as early as 7 to 10 days after conception. So when the our lady has got pregnancy, so within 7 to 10 days of conception, this HCG hormone can be detected in urine sample as well as serum sample. So it detects the presence of HCG in urine specimen at the sensitivity of 25 micro international unit per ml. So this is the sensitivity, that this much amount, even 25 micro international unit per ml of the HCG can be detected by this specific method. So HCG card pregnancy test. Here is card test. Can you mute yourself, student? Yeah, you can ask me question at the end. Thank you. The test utilizes a combination of monoclonal and polyclonal antibodies to selectively detect elevated level of HCG in the urine. Here you can see that in this procedure, the method is based on the combination of monoclonal and polyclonal antibodies. What is monoclonal antibodies and what is polyclonal antibodies? Monoclonal antibodies are those antibodies which contain only one type of antigen binding site. Okay, so it can bind to only one type of antigen in intergenic determinant. Whereas polyclonal antibodies are those antibodies, it's a mixture of antibodies. It can recognize a different antigenic determinant in the antigen. So here we have the combination of two antibodies, monoclonal and polyclonal antibodies, which selectively detect elevated level of HCG in the urine. So when there is a small amount of HCG in the urine, so these antibodies can be able to bind with HCG, and we can get the result as a some band pattern. So HCG card pregnancy test shows no cross reactivity with the structurally related glycoproteins such as the hormones such as human FHS, human LH, or human TSS. You have already studied the FHS, LH, and TSS of human origin, and here HCG also, somehow structurally somehow related to the this these hormones. But when we do this test, HCG card pregnancy test, so there is a no cross reactivity with the structurally similar hormones which are FSH, LH, or TSS. What does it mean? It means that if there is a elevated level of FSH, LH, or TSS, so it will not bind with the antibodies which are against the HCG. So HCG is very specific, hormone, and the specific antibodies, basically the monoclonal and polyclonal anti-HCG, can only bind to the HCG hormone, not to the FHS, LH, or FTSS. So there is no chances of cross reactivity with the other hormones. What is the principle? The test utilizes a combination of antibodies, including monoclonal HCG antibody, to selectively detect elevated level of HCG that we have studied. And the HCG is conducted by immersing the test strip in a urine or serum specimen. So here we have taken urine sample, and we can also take serum sample. And what we can do, we can just immerse the test strip, the pregnancy strip, into this sample. Okay, so after that, the specimen, basically the urine, when we immerse the strip in the urine, then the specimen migrates or move by a capillary action along with the membrane. Means that there is a membrane into the strip. So the specimen, what we call urine, it usually migrates or move through a capillary action, and it react with the color conjugate. This is the color conjugate means enzyme level, antibodies are there. So enzyme level antibodies, or we can say the colored level, colored compound bound antibodies are there. So it can bind with the antibody which are attached with some colored compound like enzyme. So positive specimens reacts with the specific color antibody conjugate to form a colored line at the test line region of the membrane. Here, when there is a positive specimen, does it mean a positive specimen? You have to understand that positive specimen means there is a presence of HCG in the sample. Okay, so when there is a positive specimen, it means it reacts with the specific color antibody conjugate, and after that it produces a colored line. Where? At the test line region of the membrane. So there are already their labeled line. So at the test line of the region of the membrane, usually we see the colored line. If there is absence of this colored line, it means it suggests the negative results. To serve as a procedural control, a colored line will always appear in the control line region. So other than the test line, there are control lines, and in the procedure you can see the color line always appear. So it indicates that the test strip is working properly, or it is give the validity of the strip, right? Now, specimen collection and preparation. A urine specimen must be collected in a clean and dry container. We can collect urine specimen uh, in a dry container, clean container. Usually, first morning urine sample is preferred since it generally contains the highest concentration of HCG. However, the urine specimen collected at any time of the day may be used. So, what we understand that urine can be collected any time a day, but preferentially we can do this test in the first morning urine specimen because the concentration of HCG is very high at the time of morning urine sample. Urine specimen exhibiting visible precipitates should be centrifuged, filtered, or allowed to settle to obtain a clear specimen for testing. Sometimes, usually, female may have a infection in the urinary tract. Okay, so because of the urinary tract infection, the urine that is given by the female may show turbidity, may show some precipitates. So at that time, if you want to do the pregnancy test, so in such condition, we need to do the centrifugation so that we can remove the turbid part, or we can filter it, or okay, or we can allow them to settle it so that we can obtain a clear specimen. So the clear supernatant can be taken, or the filtered can be taken for doing the pregnancy test. Urine or serum specimen may be stored at 2 to 8 degrees Celsius for up to 48 hours prior to testing. Let's say there is a delay in the testing of this test. So what we can do, we can store the urine sample or serum sample at 2 to 8 degrees Celsius for up to 48 hours prior to testing. So up to 48 hours we can do the test. For prolonged storage, specimen may be frozen and stored below minus 20 degrees Celsius. So if you want to store, if you want to do the test for after a week or after a month, then the prolonged storage can be done at minus 20 degrees Celsius. That is a storage temperature. What are the materials required for performing the test? We require timer sometime, or must of we have a mobile phone, we have a timer inside. So timer is just to look after that because we need to observe the result. We need a specimen collection container so that we can collect the specimen as a serum or serum or urine. We need a pregnancy strip or card. Pregnancy card we require, or pregnancy strip is required. And what is the procedure for this? So procedure is just first of all you have to bring the test strip and a specimen to room temperature prior to testing. At room temperature, that is what this is considered 15 to 30 degrees Celsius, or 25 plus minus 5 is the room temperature. So you need to ensure that the serum or urine sample or the test strip, the reagent, whatever you have, they all should be kept brought at the room temperature. Now, the first step in the procedure is just remove the test strip from the seal pouch and use it as soon as possible. Now, because the strips are available in the seal pouch, so you need to just tear the pouch. You need to remove the strip, and now you can make it ready for use. Immerse the test strip vertically in the urine or serum specimen for at least 10 to 15 seconds. Now, this is what you need to just strip, you need to immerse the strip vertically in the urine sample, and then place the test strip on non-absorbent flat surface. This is after the second step. When you have done the exposure of the test to the specimen, then after that you can place the test strip on a non-absorbent flat surface. So put on somewhere, maybe in the some uh, bed plate or somewhere else where the surface is not absorbent flat surface. Okay, and now start the timer. Wait for red line to appear. So timer, we'll start a timer, and we can see that at what period of time we can see the red line. Read the result within 3 minutes when the testing a urine specimen. So within the 3 minutes of the start of time, we have to read the results. For urine sample, 3 minutes, and for serum sample, you have to read within 5 minutes. Okay. So this is the timing given. This is the interpretation you can see. Now positive results. What is positive results? We can see two distinct red lines appear. So this is the strip, and this one is a control line, and this is a test line. So here we can see the control line and test line. There are there are two distinct lines has appeared. So we can say there is a the test is positive. One line should be in the control region, C region, and another line should be in the test region. Okay. Note, the intensity of the rate in the test line region will vary depending on the concentration of HCG present in the specimen. So here you can see the test line intensity means the color, the darkness, the dark color, the dark band, the dark line in the test region may vary because of the variation in the concentration of HCG in the sample. Therefore, any shade of red in the test region should be considered positive. So any line, any type of line, either light line or dark line, which appear in the test region in the strip, so they are considered as a positive sample. Now, negative sample, okay, negative for the test. How can we consider negative for the test? So one red line appear in the control region. So this is the image. Okay, we can see that there is only one line is there, which appear on the control region, seed region. In the test region, there is no line. No apparent red or pink line appear in the test region. So the test region has a no line, no any red or pink lines are there. Okay, so here we can give the negative results. Now there can be the invalid test. What is invalid test? That we can say that there is no positive, no negative. We cannot understand what is the what happened actually. We can see the invalid test as because of the expiry of the test strip, or something which happens, like error has happened. Here the control line fails to appear. Here we should have the control lines, but here control line fails to appear. In such condition, we can say the test is invalid, where we can say the test is not working properly. Okay, the strip is not working properly. Insufficient specimen volume or incorrect procedural techniques are the most likely reason for the control line failure. Here we can understand that why the test is invalid, because of insufficient specimen or incorrect procedural techniques. Okay, so the procedure that we are doing is not correct. So in that condition, the control line may fail to appear. Note, you just note it down that review the procedure again and repeat the test with the new test strip. You need to repeat once again. Okay, if the problem persists, again you have the same observation, then you need to discontinue using the test kit. Okay, and contact your some local distributor or something, because the test strip that is supplied by the distributor, you need to contact it that the kit is not showing control line. It's not a correct kit. Okay. So that local distributor will replace the test strip with the good one. Okay. This is the sensitivity that the test qualitatively detects. The test qualitatively detects the presence of acids in urine specimen at the sensitivity of 20 micro international per ml for urine and 10 micro international ml per ml for serum. So this is the sensitivity. Means minimum 20 microliter micro international unit per ml. Okay, can be detected in the urine sample, and in the serum we can detect 10 micro international unit per ml of the HCG. Okay, this is the sensitivity. These are the reference. You can go for there, and we have some animation also. So I am going to put some animation link, which you can read the this animation. You can watch the animation. Okay, this is the animation as a reference. So do watch the animation and so to understand the procedure and the principle also. So whatever we have discussed is given in the animation. Okay, I'll just share this animation on your Google Classroom. Now, if you have any question, you can ask me. We have done the discussion on principle, procedure, sensitivity, everything. If you have any question, you can ask me.